Sugar Rush is a platform that gives the campus an opportunity to come together informally and share their recent research pursuits. The aim is basically to increase campus dialogues across domains, departments, specializations, to provide opportunities to understand the very big diversity of science and innovation happening in the cluster, and promote collaborations, hopefully, so that we as a cluster can take more of science to society. Just a few housekeeping rules. So we always have two speakers. One is an academic researcher from the cluster and one is a startup and innovator from CCAMP. Both speakers have usually speak in the talk format, although today seems to be a little diversion from that. And we speakers have 15 to 20 minutes each, followed by 5 minutes of Q&A. And to prevent distractions to the speakers, unfortunately for all of you, ice cream will not start until the talks are over. So this time, while we were organizing the January edition, it just serendipitously so happened that there are two neuroscience talks that are lined up today. And we have a lot of neuroscience. Neuroscience is a hot topic at CCAMP for a lot of people, a lot of colleagues. And on that note, it really is, quite a few of my comms colleagues, for example, are from neuroscience background. So on that note, if I can ask Dr. Taslim, so it's okay, we'll move on, we have in the interest of time. So the first speaker of the day is Laina Emmanuel. She is the co-founder and CEO of BrainSightAI. BrainSightAI is a health tech startup that uses deep tech for functional brain mapping for the diagnosis and prognosis of neuropsychiatric disorders. They have recently won the Economic Times Top Innovator Award in 2024 and becoming the 6th such CCAMP startup to win that award. And is also coming on it's riding on the top of the fundraise wave, which she will talk more about. And Laina has 15 years of experience in health management, policy and consulting. She holds an MBA in healthcare from the Indian School of Business. And she has extensive experience in consulting with government organizations and also with highest impact organizations like the Clinton Health Access Initiative. And today she will talk about AI and 3D connectomics, enhanced connectomics, redefining brain diagnosis. With that, I pass on the mic to Laina.
Thank you so much. It is an honor to be here. I think I'm not a scientist, as you all know, so it's forgive me if I'm not talking too much of the science. That's my co-founder's specialty, really. So if I say anything wrong, it's my mistake. If I say anything right, it's what she has done. So let me just give you a very quick introduction to what we do, and me and my co-founders. So we started BrainSightAI 5 years ago. This was in 2019 at the peak of COVID. My co-founder had done her PhD in neurosciences from NIMHANS, where she was focused on the use of brain indicators, specifically fMRI-based indicators, for helping track psychiatric disorders. So she was working under the psychiatry department, where she focused on schizophrenia, bipolar, OCD, and worked with a large variety of data sets, neurogenetics, neuroimaging, I think she also worked with blood data to be able to figure out, and this is a very common, it's a long-standing problem in psychiatry, which is what are the markers that you can use to help patients with psychiatric disorders. There are no body-based markers like you have in kidney functioning or you have in fevers or any of those. So a lot of how psychiatry is practiced is based on psychological tests, and it is very relevant because for a doctor they need things which they can do fast and understand what is happening, but it's not really based on a body-based marker. And this has been a century-old, decade-old problem, and only recently, because of high computational power, which has been, there's a whole wave of computational work that is happening right now. And her PhD was on how do you use fMRI indicators for psychiatry. And she had some fantastic papers published in Nature where she was looking at fMRI indicators for schizophrenia. So that's how we started our work. I come from a background, as Devashi just mentioned, I come from a background of public health and MBA, and before that I used to do operating system design at NetApp, and before that Infosys. So a very varied career, and so having worked with the government and having worked with the National Institute of Health and Family Welfare, I have a very public health background, and for me, mental health was a problem that was unsolved in public health. So the moment I met Rimjim and I saw her work in this space, to me it was like, hey, this is such a great problem to solve for. I don't have entrepreneurial experience, my co-founder doesn't have entrepreneurial experience. All we knew was this is an interesting problem. It's an interesting problem scientifically, it's an interesting problem technologically, it's an interesting problem from a public health perspective. So I think my mother had told me that if you're going to fall, you should fall from the rooftop instead of aim for the moon, and you'll at least fall from the rooftop. And we were like, hey, we don't have anything to lose. Let's try and use this research and see whether it has clinical implications and whether we can actually get it to doctors. So that's how we started our journey. It's a good thing we didn't know much about entrepreneurship, because we would have realized very early on that we were one of two worldwide companies trying to do this. And the other company is Australia-based company, which got the world's first FDA for medical connectomics last year. But when we started, thankfully we didn't know that, because if we would have known that, I don't think we would have ever tried to even solve this problem. So we started, and I'll now just give you a couple, I don't want to get into the images, as she has very clearly told me, no images, nothing, chalk talk. Connectomics is really about looking at functional and structural connectivity of the brain. We use 3 data sets, your MRI data sets and more advanced MRI. So your structural MRI, which gives you the structure of the brain, your DMRI, which gives you the water diffusion in the brain, so that helps you understand the fiber tracks. And the last is your functional MRI, and within that a particular one called a resting state functional MRI, which in a resting state shows your blood oxygenation. I'm sure a lot of you must have already worked with this kind of data set. The challenge with resting state functional MRI is that there are hundreds and thousands of research papers around it, and it's been shown in research to have so many implications. But when you actually try and get something out of research into clinical setting, the biggest burden and the biggest hurdle that we had was, how do you show it's reproducible? How do you show that for a patient, it's actually making a difference in a clinical setting? And that was a really hard problem to solve for. I think when we started out, 3 months after we got our first round of initial pre-seed funding, COVID hit, and we had just about set up an office space at that point of time, and there were 3 people in that office. And COVID struck and we went offline immediately. Now, a lot of these hospitals were not focused on neuro disorders at all. Everybody was moved towards COVID and respiratory disorders and all of that. So when we started approaching doctors and hospitals saying that we have got this amazing technology, would you like to see it being translated into clinical work? And if yes, would you like to work with us to actually prove its efficacy? Not a single doctor at that point of time had the time or space to give us that. But so we also, at that point of time, BrainSight has also been working on it was our way to ensure that we survived during COVID, which was we started also working on digital phenotyping. So the vision that we then kind of put together at that point of time is psychiatric disorders have no markers. fMRI is possibly the closest you can get to some sort of marker. but fMRI reproducibility in a hospital setting is a question mark which will be solved only with thousands and thousands of data sets. How do you still, without that kind of validation, large-scale validation, still make use of it in a clinical setting? The way that we thought about is how do you kind of use fMRI, digital phenotyping, and social data and psychological data together to be able to help a patient. And I think in the beginning, and I think this is a conversation which you might be very aware of, being scientists, you're very particular about accuracy levels and sensitivity and specificity. And we used to go to doctors, and interestingly, we used to go with the specificity, sensitivity of the work Rimchim had done in schizophrenia to show that we can do something at least. And doctors used to be like, a lot of doctors were like, listen, I don't care if you don't have some of the problems that they were trying to solve for was so intense for them that they didn't care whether we had a very high accuracy or a very high sensitivity. All they wanted was that kind of information along with everything else that they had. And they were like, okay, now with this information, I will take the decision. So what we started doing was the big problem that they didn't have a solution for at that point of time was, today when a patient goes for a surgery, a lot of the, when a surgeon has the brain open at that point of time, he has very little information about the functions of the brain and what he might be affecting when he's cutting a single part of the brain. That functional map of the brain is just not available. So any information we would have given, even if it was not super high accuracy, they were like, listen, just give me anything, because for 95% of my patients, I'm not getting that. For the 5% of the patients, they were able to go for a thing called a task-based fMRI. I think you might have heard of it, where a patient goes into an MRI machine and they do a task. They either read a paper or they basically clap their hands or do some sort of thing. And when they do that, the MRI signal of that particular function is caught. Now, a task-based fMRI is great in a research setting because you have very controlled conditions. But in a hospital where the MRI department has very little time, a task-based fMRI which takes 40 minutes to map one function is absolutely not possible. It was only in rare conditions for very specific brain tumors or very specific cases where the doctors knew that, hey, around the brain tumor, the language function is for sure going to be affected. That's when they used to say, okay, go and do a task-based fMRI. The radiology department might be able to give you 40 minutes of the MRI time so that you can do something about it. But for the rest, 95% of the patients, they were not able to do a task-based fMRI. It was not economical to do a task-based fMRI, or they were too old, or they were too young, paralyzed, comatose, they couldn't do a task in the MRI. For all of them, there was no way to get to the functional map. And hence, the accuracy and the sensitivity they knew that we would get more accurate over time, but they were okay with whatever we were giving. And so I think for us as a startup, that was super important, because in an early stage, when you are in COVID times, when nobody is really giving you any data to increase your accuracy, it was important to find a problem that could be solved today and was still a very important problem for the doctors. Today, I'm very happy to say that the connectomics map, which we do, and which is also used to map the functions, but it's also used to do a lot of other things that I'll get into, is being used by over 40 hospitals. Neurosurgeons are using it for a lot of cases. They do this connectomic mapping, a resting state fMRI, to be able to map the function for brain tumor, Parkinson's, epilepsy. All of these cases, just before they do a surgery, they start using it. Now, this is in the surgery department. That's how, and I can go a little bit. I don't know how scientifically, how much depth I should be going into the structural and the functional connectivity, but I will, I'll talk a little bit about it. So I told you we take 3 data sets. Those 3 data sets are then used to map the structure. the white matter tracks. The white matter tracks are then, we basically co-register it with the brain of the patient to make it personalized, because the tract might have moved due to an accident or for whatever reasons. And at the end of the tract, we show the functional connectivity. So we're essentially showing the entire connectome. Here is the structural way in which two parts of the brain are connected to each other. And here is the functional aspects of the brain which are coming together with the structural aspects. So think of it like the wiring and the actual function. So we're able to map all of that out, and we give that. Now that's at a personalized level. So this hyper-personalized brain maps is super useful for surgery. Now they have, now you all must have seen fMRI related papers. You must have seen that it's also one of the earliest indicators for dementia, much earlier than eye scans. So what we are now moving towards, and that's a more of a group level project. So in a surgery case, you need a personalized brain map. You just need that one person's data and you can map it for that patient. But when you're looking at early signs of dementia, what you're essentially looking at is comparing data across 50, 100 patients and saying that here are the connectivity patterns, which is showing that this is most likely early signs of dementia, or this is most likely early signs of psychosis, and this is how you could use it. So we are now starting, now that we have the confidence of the hospitals, that took 5 years for us to build. Once we have the confidence of the hospitals, they have seen that we are actually making commercial sense, scientific sense, and technical sense to them. They have now adopted us. And now the neurology department and the psychiatry department, which 5 years ago we had approached them and they had said that, hey, this is too futuristic for us. We are not going to, even though there were some cutting edge papers that Rivjim had published as part of her work, they were like, this is too fancy. This will come in 7 years. Everybody was very supportive of us. They were all like, hey, this is amazing work. You will certainly make a mark in the world, but I will adopt it after 7 years. And we were like, okay, sir, by the time we'll die out, if you can you adopt us now? And they were all like, no. So that was for neurologists and psychiatrists at that point of time. But the moment the neurosurgery department in the hospital has seen its value, the neurology and the psychiatry department today have started sending us a lot of their data. They have now come to understand that even in neurology and psychiatry, personalized brain maps could help, and that if they send it to BrainSight, we can actually do group level analysis across those data sets to be able to tell them information about early signs of dementia and early forms of psychosis. So that's where we are at today in terms of where we are as a company. As I said, we are one of 2 worldwide companies, so the world is our oyster, and we need to figure out how to kind of get there. We are now in the process of applying for our FDA, and where we have to show deeper levels of reproducibility and show analytical claims and show the proof of our analytical claims. We've got our Indian licenses already, and that has allowed us to work with Indian hospitals. It's called the CDSCO, you must have worked with that. So we've got those licenses, we've done our ISOs and all of that. Now we are also aiming for the FDA, and from there we do hope to kind of be able to service a lot of hospitals across the world who till now never had access to connectomics, because usually connectomics happened in a research institute or in a Harvard or a Stanford, because they had a bunch of neuroscientists who could work with the doctor. And when a doctor says, for this particular case, can you do like a connectomic analysis and give it to me? The team of researchers and scientists could take out time and give it to that one patient and do it. But most other hospitals in the world don't have access to these kind of scientists. And I think it's something that maybe you can also think about. I think a lot of us really care about making an impact in the world, and I think the healthcare sector has a lot of space for science-led innovations. It's a hard journey because you have to get regulations right, you have to get so many things right, but it is also a highly fulfilling journey. I think the first time I got a brain surgery case in my WhatsApp from a doctor, and I had like these these open brains in my WhatsApp, and I was telling my husband that this is like, this is so crazy, because I've never ever actually been, like, I have doctors in my family, but I've never really kind of seen how they do surgeries. But I had a doctor sending me the cases and saying that, can you get the connectomic analysis done, because this patient really needs it by tomorrow. And that was such a great moment for us and for our team of 30 people who are now spread across the country, and they're all very excited. They are all very open to the idea that their WhatsApp is filled with brain tumor images, but they're very, very excited about the kind of impact that they're making in life. So with that, that's just a quick snapshot of where we are at and the journey that we're at. I'm happy to answer any questions or anything else. Yeah.
Thank you so much, Laina. Questions? Yes. Okay. Why doesn't Hiyaa go first, and then I'll pass
the
on.
students or yeah.
Okay.
Since your disease model is coming from connectome, which is unique and personalized, what are your challenges in CDESCO as well as when you're going to FDA?
So that's a great question. So when I think CDESCO, the disease models, okay, I'll tell you. When we had to show that the brain maps that, so okay, let me rephrase this thing. When you go to a CDESCO, you have to show a clinical claim. Now the clinical claim is not that we have a very high accuracy around mapping the tumor, etc. That's part of it. But the clinical claim is, if this information is available to a doctor, does that lead to better decision making? Now, one way to prove that in a study, which is different from a scientific study, but it's a clinical study, is to show a control where you're giving a set of doctors this information and another set of doctors who you're not giving this information, and then figure out how does their quality of decision making change. Do they ask better questions? Do they get a better sense of the functions of the brain? Are they? That's one set of clinical claims that we are making. The second set of analytical claims that we are making is really about the fact that even though this is not a super highly accurate connectomic profile, because connectomics is still an evolving field. I think only recently there was a 232 cell organism for whom the entire connectome was mapped. So we are hundreds of years away from mapping the whole connectome of the human. But this information, what we are claiming and which we had to prove as part of the CDESCO, was to say that whatever information we are giving is comparable to the task-based fMRI or the direct electrical stimulation, and that it lies within the periphery of that. Now, as long as you're able to show that it's within the periphery of that of the alternative, which is DES or task-based fMRI, the rest of the clinical decision making is taken by the doctor. So the fact that we are giving him this information, he knows that it is within a particular bound which is equivalent to that of the alternative. That is enough for better quality of decision making, and that is what we had to prove, and we had to prove it using multiple studies as part of our information. Now, when we go to FDA, the biggest challenge I perceive, and we are starting to work with FDA consultants right now, is a lot of the connectomic mapping and the basic atlases that we use are based on the Human Connectome project, which is based on European data. And now, a lot of the questions that we started getting very early on from CDESCO was, how do you know that this actually applies to Indian data? So we had to collect a lot of Indian data and show this equivalence that I said. Now, when we go to FDA, we need to be able to show that it also works for the American data. So there's going to be a certain level of showing this, that this is equivalence, and the way that we have designed the software is able to map for different brains and map for different brain maps. That part is going to be one of the more complex studies that we'll have to show, in addition to everything else. We'll have to show clinical validation, we'll have to show that our analytical claims are correct, we'll have to show that the usability interface of our software actually is understandable by an American radiologist and an American neurosurgeon. So these are the 4 types of studies that we'll have to show as part of the FDA as we go forward. So all of them are tough. We'll have to find the correct hospitals, we'll have to find the correct, the hospitals with the correct profile of patients, do the study for a long period with the data requirements that they have. So hard, but we'll see. Yeah.
So are they asking for prognosis? They are saying just decision making.
Correct.
Will you be asking for prognosis?
So we don't make that claim today, even though we do know that there is a lot of use of fMRI in prognosis, precisely because working in a hospital, you need the FDA stamp and the CDSCO stamp. And you're also working with the understanding that a lot of doctors feel threatened by AI and computational neuroscience and everything, right? You don't want to, I personally don't want to set ourselves up for failure by saying that we are helping in prognosis, which is why the claims that we make is all it does is it improves your clinical decision making. Rest we will leave to the doctor, even though we can go 3 steps forward. We don't. We limited ourselves to doing only that much. So yeah, thank you. Yeah, you had a question.
Yeah, so I think I can discuss later if there is time to stay now.
No, there is no time.
Okay. All right. So I'm just curious, and again, just curious, since you're looking at all these data, the doctors are looking, is there a very distinct pattern of either the structural or the functional fMRI for something like schizophrenia versus depression?
Yes. So in fact, functional MRI for schizophrenia particularly, that's what Rimcham's Nature paper was about, where it showed that the connectivity patterns for a patient with psychosis, there's an overall change in hyperconnectivity and hypoconnectivity. So essentially for patients with psychosis, that's different from your normal patients. And so you can use that to be able to guide your neurostimulation and your cognitive rehabilitation plan. So that's part of the work that we're doing with psychiatrists right now. So there is.
So if you compare for a depression, which is not psychosis.
Correct.
And schizophrenia, which is psychosis.
So depression is a lot more, we actually started with serious mental disorders. So okay, when we started talking to psychiatrists, I also had psychiatrists who gave me, I think the longest phone call I've ever had in my life is a 4-hour phone call with a psychiatrist who was very particular about the fact that in your serious mental disorders and the more diffuse mental disorders, in the more diffuse mental disorders, you need to be able to look at other data to make any sort of conclusion in a clinical setting. Which is why we did not go down the depression path yet. We are only focusing on disorders like schizophrenia, bipolar, OCD. At some point of time when we also have our interface, we are building a digital phenotyping plus a way to work with the data that psychologists actually take for a patient with depression, the social indicators really. When we find a way to put all of this seamlessly in the hands of the patient and the doctor, that's when we'll get into depression, because till then I'll get 4-hour lectures. So yes. Yeah. Any other questions? Yes, please.
Can you hear me?
Yes, yes.
Okay. Does your technology help in mapping developmental markers in the brain, for example, from embryo till adulthood and maybe beyond?
So we have only focused on beyond 18 because, in fact, below 18, the brain changes so quickly that there are 2 reasons actually. So when we try to translate it into the clinical setting, showing below 18 requires a whole set of different ethical committee approvals. Now we have not started, even though there's a lot of research which shows that it is very useful in neurodevelopmental disorders, but we'll build up to it right now. So we want to be able to show it for adults before we start getting into children disorders. Right now we're working with the Tata Memorial Hospital where they're actually sending us cases for brain tumor in children, but we provide it as a service but not as a product yet, because as a service we are able to work with them and actually kind of map this for the children who they are operating on and ensure that there's no functional loss, but not yet as a product, because it will take much longer time to get the CDESCO approvals for working with children below 18 years.
Thank you, fantastic talk. I know time is not on such, I'll be brief. So, considering that this has been built and being used as well, at least in Indian scenario professionally, if you could possibly share an example of how this actually made a huge difference in a particular case. It would possibly allow many people to connect with the larger picture as well, because and the case study where you could briefly say it will be fantastic. Yeah.
I'll be happy to show pictures if you're okay with it. Else I will. Okay, let me just. So this is the language connectome. So we are able to map 9 different networks of the brain, which includes your language, sensory motor, your fine sensory motor. We are also able to map things like your default mode network, attention network, all of those networks. And this is what we then send across to the hospital. Now, I'll just show you some of the cases where, right. So this was a craniotomy planning for a patient. Now in this case, they did, you can see the tumor. I'll actually show you the next one. You can see the tumor, and you can see that the language connectome is actually pretty close to the tumor. Now, usually what doctors would have done is they would have opened up the brain. If the patient, the 10% patient had given the permission for awake brain surgery, they would have evoked it then. Or they would have asked this patient to go for a task-based fMRI. But the patient might not have done, and very few hospitals in India actually have a task-based fMRI because it's quite expensive. In our case, they sent the patient for a resting state fMRI, and you can see that we mapped out the language function connectome around the tumor. Now, with this information, the doctor is then able to make decisions on whether the patient's brain has to be cut open from the back, or it needs to, or will he approach it from the side, or will he approach it from this side. Now, there are also now doctors who have gone for advanced surgery techniques in the US, where when they're doing a brain tumor, they don't open the entire brain. That's where the world is moving towards. So they do just a single cut over the forehead, and they are putting in a probe into the brain, and they are using that probe to be able to get to the tumor area. Now in such cases, awake brain surgery is totally out of the picture. Your task-based surgery also is not something that they do. In such cases, what we are able to do is to actually show them this connectome and map it. Now, the validations that we have done in hospitals, I'm so sorry for all the blood here, though you might be a set of people who are okay with it. I think a lot of people, when I show this, they don't like it. What we are able to show, as you can see here, is we are able to map the fine motor areas. We map this and give it back to the doctor. And as you can see, when they're actually doing the stimulation here, this is, sorry, the patient's limb, like basically his hand moves. And so this is how they are using it for a lot of the stroke rehabilitation cases. And even after brain tumor, if there is some sort of functional weakness in the patient, they use these maps to be able to make it better for that patient. So these are some of the ways in which doctors are using it. And there are lots more techniques that are now, I think connectomics is a very starting. There are hundreds of things you can do over connectomics which are still to be explored. And I'm hoping some of my competitors will come from here. So thank you.
Okay, thank you, Laina, for that amazing talk and for those amazing results, and congratulations, and so happy to see such a deep tech startup, making so much progress in the clinical world. That's good news for Indian healthcare for sure. So our next speaker today is Dr. Hiyaa Ghosh. She is an associate professor at the National Center for Biological Sciences. She's also a global an EMBO global investigator and a senior research fellow at the Wellcome Trust, DBT Wellcome Trust India Alliance. She holds a PhD in molecular genetics from the University of Pittsburgh, followed by a postdoc in immunology from the University of Columbia. And then she did a short fellowship, which probably changed the tide. A short fellowship in the Department of Neuroscience at the Albert Einstein School of Medicine, where she worked on brain regeneration. Currently, her lab at NCBS focuses on adult brain homeostasis and neuroinflammation. And specifically, they look at genetic regulation that goes into neural plasticity, which is so crucial for so many brain functions. And also, they look at the deregulations, which could be the basic root cause for neural pathologies. So with that, I think today her talk is neurons in the adult brain, dynamics and diversity.
Thank you, Ghosh, and all of you for showing up here. And a big round of applause to Laina again. Bravo for making it work against resistance, and fantastic to see, all the functional. And so to all of you, I'll request to kind of dial it down in terms of Zoom. So what she showed here was tracked, right? So it's like a collective, we're talking about perhaps even few millimeter kind of thing. We're going to dial down 100x maybe, go into cells, perhaps also molecules, and also conceptually what she talked about is a nice demonstration of how science that we have learned, whether even if it's in clinical context, is made use for human welfare. But then if you take it few deeper notches, you would perhaps appreciate that we also need to continue to generate knowledge about how fundamentally things work, right? So that is where from this talk is going to come about. So my lab is focusing on understanding brain resilience. So resilience, if you may, is essentially the inbuilt property is flexibility to adapt, right? So if we are talking about brain resilience, what we are trying to understand is what is the scope there? We're talking about an adult brain. Neurons are formed, complicated circuits, the structure that they talk about is this white matter tract. It's all formed, and now we are to live a lifespan. And so what is the scope of adaptability here? What's the scope of flexibility? And so, if we are to forget about the board maybe, so if you had to talk about what is the scope, then we start with the components, right? So we have neurons. So mostly what this is showing up is a biased view of neurons, and the rest of the brain is empty. But in reality, that's not true. But let's focus on neurons, right? So neurons have the cell body somewhere, and then a lot of dendrites kind of coming along. So dendrites is the physical conduit through which the circuit is getting connected. But then on top of the dendrites are these little dots. All right, these are the spines. This is where synapse happens. This is where connection and transmission of information is happening. And this is the structurally modified, flexible part of the brain. Whenever we're talking about plasticity, whether in the scale of milliseconds or years, lifespan, whatever, the most amount of studies we have, or the most amount of understanding we have, is to do with the activity, pattern, activity, amount of one region versus another, in terms of which at the functional level comes to the substrates these, all right? But then, I think here what I'm trying to emphasize is that there are two things where there could be scope. One is the spine, which is already structurally flexible. One is the dendrite. But the dendrite's ability or scope of flexibility is a very understated topic. In fact, what is thought is after the postnatal period of readjustment of circuits and all, it's pretty much done and it doesn't change. So we're going to talk about that, what we learn, can we learn more about it, and would that hold importance? But then, like I was saying, the brain is not just neurons. There are plenty of other cells. So this is why I needed the images. So, right. So this is a cartoon image, and you can see there are ependymal cells. These line the blood vessels and carry a lot of systemic information to the brain parenchyma. Yeah. So that is there, but inside the parenchyma there is oligodendrocytes which are wrapping up the axons of the neurons with the myelin sheath. There is astrocyte, there is shabby-looking microglia, they're usually prettier, and then astrocytes and so forth, right? So there are a lot of other cell types which may also confer the scope for flexibility. And in that, I also want to emphasize that the things get more complicated when you zoom in, which is that each of this, as is, there is neuron, there are many types of neuron in different parts of the brain, and even within a type there is different structures and also. But then when you talk about glia, glia also has many varieties. Okay, so if we focus on just microglia, for instance, a cell type that we also work on, there is a variety of microglial state that has been understood now. Now with the advent of technology such as single cell transcriptomic, we can actually look into the gene expression profile of every single cell of a certain type. And what we learn is that there is enormous variety of the gene expression profile. What that means in terms of function is the job going forward now. And within that, if I talk about microglia, for instance, in the healthy brain, there are many types. In the unhealthy brain, there are many types too. If you have fever, the microglia type is different. If you have injury, it is different, right? So essentially what it is saying is that the terminally differentiated adult cells seem to have a lot of possibilities, right? If we learn more about what are the possibilities, how are they functionally related, and how may we be able to regulate it, perhaps we'll know the scope of adaptability more. And then on top of all this, there is also this really nice thing about the adult brain is that there are stem cells in the adult brain, okay? Neural stem cells. Can make neurons, can make glia. Fantastic. But unlike, and this is, I also wanted this, I dropped also. So this red area, so when I'm saying that there are adult neural stem cells in the brain, doesn't mean that they're just thrown around everywhere and then you can just regenerate neurons, right? But that's not the truth. The truth is that they are only located in really small areas of the brain. One is the lateral ventricles, and the other site is tucked deep inside the hippocampus, is the human brain, and this is the dentate of the hippocampus. If you zoom in, this is what's going on. A lot of neurons already there, granule neurons, pyramidal neurons, whatnot, from the embryonic time. Okay, these are developmentally born, fully functional, everything. On top of it, turns out that these stem cells line the first layer of this place and continually make neurons. Okay. Now, we have known that stem cells in adult tissues exist. And in many instances, it's been proven that the idea or the utility of those stem cells in adult tissue is to invoke them when needed to regenerate, right? So then why do we have neurodegeneration? So it turns out, unlike other organ adult stem cells, the neural stem cells in the adult brain are always functioning. They're not quiescent. They're not to be invoked because of X and Z. You know, they are continually working and they have a bona fide function. And that function in the last 2 decades have been more and more clear, which attributes it to a certain critical window of a granule neuron's membrane property, which makes it low threshold hyperexcitable. And that property has a role in the dentate circuit. So if you now want to have the contribution of this neuron, then you have to continually make it and kill it, right? Because you can only use it during the first 5-6 weeks of its birth. That is when that property emerges. So just to emphasize that there is neural stem cell, and it's exciting, it can make neurons, but it has a bona fide function that has proper role in cognition, day-to-day cognition. For example, our ability to discern between very, very similar things as a evolved organism comes from this circuit of dentate in the hippocampus, and the adult born neurons are critical for that. And similarly, there is another ability we have, which is reversal learning, our ability to kind of erase memory to rewrite. And that's a very, we talk about memory all the time, but forgetting is essential to our survival. We need to be able to rewrite to kind of move on, and there are post-traumatic disorders that people have known. And then the one important thing that these adult neurons are known for are their contribution in the stress resilience circuit. So when something goes wrong, different people have different ability to cope with it, right? And this ability to cope with the different levels of stress is actually regulated by a circuit that is between the hippocampus and the amygdala. And these adult born neurons are an integral part of it. So with the technology, we now can, in a live mouse, switch off neurons that we want to switch off and see what they're important for. And those are the kind of technologies that have allowed this information. These adult born neurons in this critical time window are required, are like essential part of this normal circuitry, right? Now I told you that stem cells are there, they make only some type of neuron in a certain circuit, so maybe boring. But no, and they're not boring, because they can tell us much more about stress resilient circuit and also. But at the end of the day, these are stem cells, right? And they have the ability to make, because we as human beings have like pioneered ways to understand what really goes on and manipulate it for our advantage. So that scope still is there. And the interesting part of the stem cell that has been discovered is that even if it's sitting deep inside one little area in the brain, it's somehow able to sense the environment of an organism. And this is like about more than 20 years old experiment which showed how sensitive these cells are to the environment of an organism. So this is a standard mouse cage where the mouse only has bedding and food. Very boring. The other one is an enriched environment where it has running wheels, tubes, can do all sorts of playful activity voluntarily. And then if you go inside and look in the brains of the standard housing, by marking, there are ways to mark only the newborn neurons. You see these many are formed. If you compare with the enriched, so many more are formed. And I just told you that these are the neurons that are integral part of the circuit where stress resilience is concerned, pattern separation is concerned, reversal learning is concerned, right? So there's got to be an influence of these stem cells to influence cognition, and perhaps we can manipulate it non-invasively if we learn more about it. And then reverse, if you put, so mice are social animals like us. If you just singly house them, one mouse in a cage, they're sad. That's psychological stress for them, and this is what happens then. Alright, so now we know that the stem cells that make very important neurons, important for stress resilience. However, when there is stress, they're not made. Right? So clearly this is a circular loop, and there is so much more to be understood, and this is one area where the lab works to understand how can we learn more about the neural stem cells. For instance, how do they sense the environment? What are the signals that convey that there is stress, or there is isolation, whatever, and when that happens, how they might modulate, what are the actual dynamics going on in the neurogenic program or in the circuit that will confer stress resilience versus not. But then that's that, and today, as she said that I was going to talk about neurons, so I'm going to go back to that. So coming back to this possibility that perhaps when we are looking about the adaptability or the scope of flexibility for neurons and only being fixated on the spines and the plasticity thereof, maybe we are missing something. And so my lab was interested to understand what about the dendritic structure. And also, relevant to the previous talk, we also started with a lot of studies with the gene that is schizophrenia risk allele. And how my interest as a basic scientist went on to that gene is not because it's schizophrenia relevant, is it because I was studying it in the immune system. And there what I found is that this gene is very important for enabling a hematopoietic stem cell, which is blood stem cell, to make a certain kind of blood cell type. Okay? However, during the life of that blood cell, you needed to have this gene continually be active and doing its function. And through, in vivo lineage tracing, we saw that just if you take out this gene, the cell doesn't die or disappear, it just becomes another immune cell type. Right? So that gives you an idea that even if it is a terminally differentiated cell type, whatever type of cell, it's not a fixed, written in stone kind of status. It can change. And there is active gene regulation that is continually added to have it be this or that. Okay? So that is where I was coming from in immunology, and I was like, oh, fantastic, this gene happens to be so relevant in the brain, schizophrenia, developmental disorder, whatever, let's look it up. So when we look up turns out, and I would have loved to show the image, but regardless, that it is abundantly expressed in the adult brain. And not just in neurons, but in many different cell types, in all of the brain regions. And then we started doing what a basic scientist does, chase our curiosity, we'll delete it in this cell type, or that cell type, or that cell type. And we could do that in contrast to the other studies that were trying to chase it for the schizophrenia and couldn't figure out what was the association. In contrast to those studies, we were able to delete this gene in neuron only, in a fully developed mouse, all right? So not a straight knockout, but a knockout that is inducible, meaning when we want to delete the gene, that is when we delete. And of course, if you want to know what it is doing in adult brain, you don't want to mess up the developmental process because it is a developmentally important brain gene. So you would let the mouse grow, all adult, all connectomes, all cell types that had to be are there, connected, everything. Then you go take out this gene at the time of your choice and in the cell type of your choice, which was neuron for us in the first instance. And when we did that, what we saw within 2 weeks is that the dendrites fully changed. And now this is where my drawing part will have to come, unfortunately. So I'm going to draw a very bad neuron here. Let's say these are the dendrites, and forget about the spines, we're not going to talk about that. And so when this is from an adult brain, and all we are doing is we delete this gene, and within 2 weeks, what we see is that there is more branching, okay, in this neuron, and therefore the action, and we also see that the action potential, properties are very different. So the neurons have become different, their structure has become different, and therefore, of course, there are cognitive deficits and so forth. So what is so exciting about it? The exciting thing for us was, again, in the same vein of what we had seen in the blood system, is this is a fully mature cell type which for decades are thought to be stable, but turns out that the intended structure, which is critical to its connectivity, is an actively maintained status, right? And which then can be inferred that perhaps this is so that flexibility is retained. If I have to become more branchy, I have that ability by downregulating TCF4. However, if I was set in stone to be this way, then I wouldn't need any active regulation, right? That would be energy conserving, but that's not how nature made it. So we inferred it as retained flexibility in adult neurons to do that. So during these studies, when we are looking at different cell types for their scope of flexibility, this is a concept that again emerged in the adult neural stem cell context also. So it turns out that the adult neural stem cells, which look like this, they are called radial glia-like cell, actually only known to make neurons and glia, actually also have a suppressive gene network going on in them, which allows them to be neural stem cell. And if you delete this suppressive network, then they cannot make neurons, they cannot make glia, instead they make myeloid-like inflammatory fractures. All right. Now this is again another mystery as to why would you have stem cell retain the ability to make inflammatory molecules, right? And so then the investigation pursues, and the hypothesis then is that inflammation may have a bad name to it, but it's really a good thing for physiology. It is the thing that flags the system that something's gone wrong, bring back action, set it all right and all. So perhaps this cell type, which remember senses the environment, retains this inflammatory potential to perhaps figure out things for homeostatic purpose, meaning bringing back things to normal. So then we are able to now see in 2 adult brain cells that a whole program of repressive network allows the cell to be what it should be, which is to say that it retains a lot of the flexibility. And we are now trying to understand the biological significance of it, right? If the neuron can change its structure, why it might be, in which case might it be, how does it happen, and also, and same for the adult neural stem cell, is there inflammation, how can we make a physiological situation? And so in this case, it was easy for us because stress resilience is conferred by adult neural stem cell. And so we said that, okay, let's look out if during stress of an organism, what really happens. So we established a few model system where we take the animal through some kind of physiological perturbation. It could be a neuronal mutation, it could be stress, psychological stress, it could be brain injury, whatever. And then we see, well, you lose cognition as a test of things went wrong. And then if you just let the mice be, put them back in their cage, do nothing. After a certain period of time, they gain their cognitive sharps, right? They're back to normal. Not surprising, but very good model for us because we did nothing. The system did whatever it did by itself to bring back normalcy. And then we had this window to kind of go and check what happened from the neurogenic program side, what happened in neuronal. And what we see for this is that actually this gene that we knocked out to kind of present a proof of concept that the neural stem cell has inflammatory ability actually goes down. And we are now able to follow the whole neurogenic program to see what then happens after that and after that and after that. And we have some good data to suggest how the dynamic adjustment of the neurogenic program, adult neurogenic program, confers stress resilience. But more importantly, in the neuron side, I think this is I really wanted to show because this is like really the exciting part of the story that's coming up. So when we were doing these investigations, we use very complicated but sophisticated genetic tools that allow us to see a lot of things that we can't imagine. And then you see it and then you can pursue it. So what we were doing is genetic labeling with a proxy reporter. So if we delete anything in any cell, we have the inbuilt design in the system to turn that cell green forever. Okay, and so we did that. This is the CA1 region of the hippocampus, very densely packed with cell, but you can see that the green is not looking so dense. And if you see more closely, there are plenty of pyramidal neurons that are not green, even though they're supposed to express the same gene through which we are deleting the dog-speaking system. And so this then showed us that there are two types in the thing. So this again opened up two directions. One is that if resilience is happening and the circuit is needed for resilience, when we knocked out the gene, we changed the structure of these one. So is it the other ones that we couldn't do anything of, which we call the bystander neuron, that could compensate for the structure? And indeed we see that, so there is this thing called shoal analysis, where we can say the structure, we can quantify the actual structure, the whole dendritic tree, and we see that when these become more branchy, at the same time when we do nothing to the non-green, they become less branchy. Alright, so this is another example where we can have some proof of concept that this dendritic structure flexibility probably plays out in real life and could be for adaptive compensatory role as well. The other situation where we find this happen is during the lifespan. So now we are talking about wild type mice where nothing has been disturbed. And turns out that the dendritic structure, so this is the shoal analysis quantitation I started talking about, it kind of changes. So the neurons become less branchy, but their location of branchiness changes with age, right? And again, a curious question as to why that might be relevant. Turns out how we encode something changes. Like if we are, take an example of space, how we encode space in our 30s is different from how we encode it in our 60s. And that is a reality of physiological process for which there must be more evolutionary reasons for why that would be advantageous. So it turns out that there probably is a whole scope of dendritic structure changes in normal health, in cognitive changes. And then something more interesting that we have bumped onto is that the other hypotheses were that maybe even if they are talking to the same structure circuit, they're probably employed for different cognitive purposes. So we did some tests, and turns out it is possible that one is more important for encoding, whereas the other is more important for recall. So there's a whole concept of engram cells which is picking up more evidence and details, and turns out that this diversity within the same population, in the same circuit, with very similar structure, just one is skinnier than the other, but branch patterns are same, could have a more deeper reason in the context of resilience, perhaps. So, I think I just ended that.
Questions?
So I just want a little clarity that the perturbation, what you are saying, because I am asking very documentary kind of a question that are they very equivalent to the life-changing events? You know, because of trauma and this. And another similar thing that to deal with stress and depression, what happens in the frontal lobe? Because I think we were not talking about the frontal lobe,
Yeah.
which shows in memory.
So that will fire again, but it has to be, mostly it comes from the ones that are fired during the time the thing has happened. And this representation has a copy of it in the, for example, in the prefrontal cortex. So your question was, what do we know about that in the cortex, right?
No, I wanted to know that is your perturbations are very much similar to the life-changing.
Okay, yeah, so that's the first. Yeah, so the first thing, for example, this I was showing, this is like as we kind of find weird things about biology, we kind of chase this down to real physiology, right? So this is one of the contexts to say that in a healthy mouse brain, no perturbation, changes are happening in the dendrites. Now, coming to the perturbations that we see to find a window, looking back to see how homeostasis is happening, we, for example, use social isolation. Happened during COVID, very relevant. We also use immobilization, which is not true for perhaps human being in that way, which what we model in mouse, but immobilization kind of stress can be extrapolated for psychological stress of other type. There is social dominant stress models where in human context also that does. Then we have traumatic models where we can do very controlled brain injuries to understand other topics. And then the neuronal mutations are really a brute force method where we want to say that, well, structure changed and then this structure can it change back if I have to connect to the same circuit, or do the other neurons then compensate to change and all. So yeah, so I mean the whole idea is to be able to relate it to normal physiological relevance.
I think this is super exciting. I just wanted to say two things which I'm taking back from this. One, which is I think we get a lot of requests from doctors who are working with coma patients as well. And there is work in how connectomes in certain areas of the brain can help in understanding if that patient will come out of coma. And your whole work around this is exciting because there's so much potential for regeneration there. And the second, which we get a lot of requests, is around understanding neuroinflammation in a number of disorders and myelination. And so both the things which you've touched on, and there's so much we can talk about in terms of how it can be translated for clinical work as well. So thank you so much.
So to that I'll say that, because of our interest in the schizophrenia risk gene, and that same gene being so relevant to the adult brain cells, we recently made 2 CRISPR mouse to look into it because I got really interested in the locus from a basic science perspective, and that is leading us to some schizophrenia kind of collaboration. We have a person, I don't know, Vijay Vishwanath, I think a lot of us know, in the psychiatry department. He's very research oriented, so we're trying to kind of bring the basics, the clinical data, and kind of extrapolate it for understanding basic science from the basic science perspective. And also for inflammation, again, inflammation has a bad name but also has a blanket use. Everything is inflammation. But I think it's important to recognize that it's inflammation, but then getting into the details of what kinds is very important. One thing that is very common in everything neurological, psychiatric, developmental, infectious, degenerative, whatever if the brain has a pathology, microglia looks activated, right? And a common course of drugs is to just dampen inflammation, like whatever it is. And it works, because generally if you want to bring back the niche to kind of in a little calmer form so that they can function normally, you would attain some bit of normalcy. But turns out that whether microglia can be deactivated or not is not known. Right? We know that microglia can activate it, but can they be deactivated? And these are the fundamental questions that we need to answer from a basic science perspective. That is also something that my lab also my brain is very involved in, where we have some really fantastic data to suggest that microglia cannot be deactivated. It has to be replaced. And the more we learn about all these various types that are there, what are they important for, when do they come, if they go back, what of that are the basic science connections that can, have all of this understood better.
I'll just go quickly. So, I was to ask something else, but just for the microglia, would one consider that rather than deactivating it, it's a state, in a way, which is because of the upstream triggers, which is actually activating it in a way. So stopping the triggers would change the state of it rather than deactivating it, and I don't know how much dynamic it is in terms of the time duration it is, right? So
very pertinent question.
Okay.
The dynamics are extremely fast. So much so that, so that others can listen, so much so that, so we do a lot of cell culture counter model, right, to understand a lot of biological processes, even though we know that, creating the whole niche is very important to understand a thing. In the case of microglia, what people have shown is, so microglia is a macrophage, right? It is a tissue macrophage. We have tissue liver macrophage, we have tissue lung macrophage, they have different names. The brain one has microglia as the name. However, this is macrophage whose fundamental purpose is to sense the environment of the tissue. And they have shown that as soon as you take out microglia from the brain and put it in a petri dish, within 6 hours its transcriptome only changes. Alright? So, and then in that form, what it retains is the basic macrophage property. And now the activation of a blood macrophage or a Kupffer cell or whatever will be different from a microglia, right? And so it is really fast. And in fact, we see that in our models also. So, catching it in action, you would perhaps be against the whole fundamental role it plays. Right? Before you know, they'll know it, the microglia. But I think there is a huge scope because they clear up on its own. So we have some single cell based, in vivo model studies where we see that when microglia activation happens, it's not pan. We see that even in the steady state brain, in a little region, cortical region, there are at least 6 types of microglia. And in response to brain injury, not all of them amplify or get activated. Only the ones that carry a certain immune profile are the ones that amplify. And then if you chase it down, they're not there after that. So there's an automatic clearing mechanism. And perhaps what we have to focus on is to figure all this out. Who comes up in response to what? And what's the clearing mechanism and where is that getting impeded? Perhaps.
So I'll ask a very layman question, so pardon me. See, the last talk which I got the impression is that there are certain regions of brains, right? And when I'm hearing you, it is coming that the brain is very adaptable, there are no regions, right? So, there are no regions, specific regions, say for language or something of that sort.
No. Yeah, so I'd like to clarify that if I gave another expression. So I think that question was specifically about memory consolidation, right? If we are doing a motor function, that has nothing to do with memory. I mean, it has, but at the time of executing the function, it's very motor. So in fact, every region of a body, it has a particular map in the brain. And this mapping, very region-specific mapping, starts right at the time of development and continues to kind of wire and rewire. So there are, for example, if I'm a violinist, I'm using my left hand, and in the left hand, these 4 digits more on the string. And if you look at the brain activity, the volume, then it is, and each of the digits have a particular mapped imprint in the brain, right? There is a neuronal neuronal representation of connecting to that particular muscle. And what you see is that for the violinist, that part of the brain which represents these 4 digits is bigger. And if I'm an older violinist, that will be even bigger. So there is a plasticity of order that goes on throughout our lifespan, okay? And it's not that it's not known, it is known. That's why we say brain is very plastic, but the details of it is not understood, right? So when she's talking about the brain tract and the activity, it's at a very zoomed out scale, right? So all we can say, yes, that happens, that has been known. Now she's trying to connect, if we know this, how can we utilize it? What I'm trying to do is from the other end, If that happens, how does that happen? What are the details? Right, so we can learn more too.
I think thank you, Hiyaa, for an amazing talk, and I hope this serendipitous aligning of stars where Laina and you came together. I hope that sparks of collaborations in future, and that's the entire point of Sugar Rush with Science. And thank you so much, both of you, and thanks everybody for listening in. And ice cream is now on.